Hello future medtechs. So our activity for today is activity number 2, the compound microscope. So objectives for today are, at the end of the activity, the students are able to, number 1, describe and demonstrate how to properly use, transport, clean, and store the compound microscope. Number 2, identify the parts of the compound microscope and give the function or functions of each part. 3. Demonstrate proper placing of a prepared slide on the stage of the microscope. And lastly, demonstrate how to properly use the different objective lenses.
A compound microscope is an expensive instrument in the laboratory. Without it, it is impossible for a laboratorian to perform essential laboratory tests. Therefore, proper care and handling is required, especially when transporting this delicate instrument. Since most compound microscopes are heavy, it is nice to know these tips on how to properly carry and transport a compound microscope to avoid accidents. Pick up the microscope with both hands, one hand holding its arm and the other hand supporting the base with the power cord securely intact. Carefully place the microscope on your work table, about 5 to 7 inches away from the edge. Before you start manipulating the microscope, you have to make sure na nakaset siya sa scanner. Bakit? Kasi ang scanner ang shortest objective lens para hindi siya mabangga dun sa ating microscope stage. So guys, let's tackle the following parts of the microscope. Eyepiece or ocular lens. Eyepiece is the lens present at the top and is used to see the objects under study. Eyepiece lens contains a magnification of 10 times or 15 times. Tube. Tube or the body tube connects the eyepiece to the objective lenses. Resolving nosepiece. It is also known as the turret. Resolving nosepiece has holders for the different objective lenses. It allows the rotation of the lenses while viewing. Objective lenses. Generally, 3 or 4 objective lenses are found on a microscope with ranges of 10 times, 40 times, and 100 times powers. Lenses are color-coded. The shortest lens is of the lowest power, and the longest lens is high-power lens. Diaphragm. Diaphragm helps in controlling the amount of light that is passing through the opening of the stage. It is helpful in the adjustment of the control of light that enters. Coarse adjustment knob, used for focus on scanning. Usually, the low-power lens is used enabling the movement of the tube. Fine adjustment knob, used for focus on oil. Moves the body tube for focusing the high-power lens. Arm. It supports the tube of the microscope and connects to the base of the microscope. Stage. The platform that is flat, used for placing the slides under observation. Stage clip. Stage clips hold the slides in proper place. Base. Provides basal support for the microscope. And then lastly, the power switch. The main power switch that turns the illumination on or off. For the next videos, your laboratory instructors will demonstrate on how to properly manipulate the microscope. In here, we examined different prepared slides of sperm cells, simple epithelial tissue, smooth muscles, and the human blood cells. This is what we see on a scanner lens. Next, we will use the low power objective lens. And then the high power objective. As you can see, your instructor only used the fine adjustment knob. And lastly, our highest power objective, which is the OIO, or the oil immersion objective. When using OIO, you have to put a small amount of immersion oil because that oil will increase the numerical aperture of your lens, which will result to a better image. Before placing the slide onto the stage, you have to determine or locate the specimen or your sample that you will examine. In viewing using the scanner or the low power objective, you can use the coarse adjustment knob. When you are viewing using the high power objective, you should only use the fine adjustment knob. You can also use your fine adjustment knob for minimal adjustments when focusing your tissue sample or your specimen. can move the stage forward and backward using the upper knob. You can move from left to right using the lower knob. Please do not forget to perform the aftercare. During aftercare, you have to wipe the immersion oil both on the objective and on the slide. Put it back on a scanner setup. Then turn it off, unplug, then voila! You're done for today. And that is all about the compound microscope.